Hello everybody, my name is Peter Klapper from the School of Biosciences at the University of Kent in Canterbury, and in this video clip I want to discuss with you why kinetics is a very exciting topic and why knowing about kinetics can cure potentially cancer and maybe even something like Ebola. So, how does it work? What is it all about? Now, instead of telling you what kinetics is or what it isn't, let's have a look at a little example. Let's say, for example, we have a cell. This is the cell surface, the cell membrane. Here is the nucleus in the cell, and the message gets out that the cell should make a certain receptor protein. So let this be our receptor protein that is made in the cytoplasm through transcription and translation. And now this receptor protein is transported at a certain speed to the cell membrane, where it is anchored in the cell membrane like that. Now we've got loads and loads of different receptors, and these receptors are usually waiting for a signal from outside the cell. These signals usually are generated by other cells, or they might be even produced by the same cell. It doesn't matter in this case, but these signals tell our little cell what to do. For example, the signal is saying, little cell, you are now old enough, go out and divide. So let's say we have a ligand here. So that would be a ligand. And that was our receptor. Now this ligand and this receptor, they interact, and the receptor binds the ligand. And we've got something like that. So we've got ligand-receptor interaction. And what very often happens is that if we've got a second receptor and second ligand bound, these things dimerize. So we might have a situation where we have something like that. And these two dimerize. And very often what we find is that it is the ligand receptor dimer, and I'll draw it like that, something like that, with two ligands bound. These then very often autophosphorylate or cross-phosphorylate, and the most important thing is that in this case a signal is delivered to the cell. So this could be, for example, cell, divide, do something, become active, or whatever. And it is very often that it is this dimer of receptor ligand. And of course we don't want this switch on all the time. So what happens in the cell is that usually these receptors then are somehow, by some mechanism, are degraded. And also the ligand might be degraded, so they are all removed, and basically the system can start again. But the important thing was that the cell has received a signal. Now, what does this all have to do with cancer, and what has it to do with kinetics? Well, the interesting thing is with kinetics is that everything has to be very fine-tuned. So, for example, if we don't transport a lot of receptor to the cell membrane, well, nothing will happen. There is not enough receptor then for the ligand, and there won't be any signal. Likewise, if we don't have a ligand and they don't bind in a good concerted manner, well, we won't get a ligand receptor. If they don't dimerize properly, if the speed of dimerization is for example, slower than the degradation process, again, we won't get a signal. We won't get this stable ligand-receptor dimer. On the other hand, if our receptor degradation, if this dimer degradation here is very fast, is faster than this one here, this process here, than the dimerization, again we don't get a signal. But if it is slower than this dimerization, our signal will be on for much longer. So the cell will constantly get the signal and the order divide now. And we all know what it means if a cell is constantly dividing. It means cancer. So by looking at these processes, we know that they have to be at the right speed. It has to be the right speed where this receptor is transported to the cell membrane. It has to be the right speed by which this receptor interacts with the ligand. It has to be the right speed where these two ligand receptor monomers dimerize to this dimer. It has to be the right speed where the signal is transmitted, and it has to be the right speed where the dimer is degraded. And if only one of these speeds is out of kilter, then potentially disaster happens. Either the cell doesn't get the signal to divide, or it continuously divides. And we have a similar situation. For example, look at the spread of a disease. So we might have, say, a disease in one person. So this is here person who has a disease, and we can now look at how this disease spreads through the population. And again, this has something to do with kinetics. How fast does the disease spread? How fast can we, for example, immunize against this disease? And maybe we can even determine which people are crucial for this immunization, depending on how fast this disease spreads through the population, how fast people can recover. So if we know all these parameters about a disease, about the kinetic parameters of this disease, we can take action and make sure that this disease does not cause too many casualties. So I hope that with this clip I've convinced you that as a bioscientist it's definitely worth knowing about kinetics, and it's not really that bad. In the next few videos I will go a little bit more into detail what it all means and how we can look at kinetic parameters. Thank you very much for watching, and I hope it made sense.